barriers. We need to transmit both molecules and information from one side of these membranes to the other. In some cases, the right kind of molecule is capable of some amount of diffusion through a lipid bilayer. And if you can see right down here, we have a graph of increasing permeability of molecules through that bilayer with highly charged ions such as sodium and potassium exhibiting very low permeability, and molecules like glycerol or water exhibiting reasonable permeability. But these, the rate of transfer, the rate of diffusion is pretty slow compared to what the cell needs, and especially with regard to transport of highly charged species like ions, the cell needs gateways. And we should be able to control the flow of these molecules in and out of the cell. So here's where our membrane proteins come in. So about 30% of all of the proteins that are made by the cell end up being associated with the membrane. And until recently, we actually didn't know too much about the relationship between the structure and function of these important membrane proteins because they were so difficult to study and to obtain high-resolution structures. So even though 30% of the proteins are in the membrane, less than 2% of the structures that we have in the protein data bank include high-resolution information about these proteins. So you'll see that these proteins can sit in the membrane. We'll take a look at different types of membrane proteins. Some of them are embedded in the membrane. Some of them are just associated with the surface. But we'll see how they can form channels that can be opened and closed, and how they can be specifically associated to allow the passage of particular ions or molecules from one side of the membrane to the other. Just thinking about diffusion of a polar molecule, we can consider what the energy would be as we push a polar molecule from one side of the membrane to the other. And if you take a look at this free energy profile, you'll see that when that polar molecule is sitting in that hydrophobic environment, right in the middle of that bilayer, that's a very high energy state. And a lot of that high energy is also associated with the fact that those polar molecules need to be desolvated. Right? So on the extracellular side, all those water molecules are sitting around that polar molecule. That's a very energetically favorable condition, but they all need to be stripped off before that molecule can diffuse through the cell membrane. So that explains this huge energy barrier. And just like any other event within the cell that has a very high energy barrier and the cell wants to be able to speed these processes up, proteins can be brought into the picture to facilitate these kinds of events. So down at the bottom, if we just embed within that lipid bilayer environment within the middle of it, that provides a much lower energy way to transition that polar molecule from one side to the other. Right? Yeah, maybe the solvation sphere around that molecule will need to be removed, but that polar molecule will still be properly associated with the polar environment in the interior of the protein. Does that make sense? So in some extent, you can think of this as a catalyst for allowing more rapid transport of a polar molecule from one side to the other. Yeah?
So in general, do enzymes that undergo catalysis just lower the energy of the transition state, or can they actually change the transition state itself and use a different mechanism?
Right. So one of the key review for the midterm, I think that's really crucial to point out that one of the ways that enzymes can facilitate reactions is to change the mechanism by which they undergo conversion from substrate to product. Okay? Yeah. Good. So here are the three main categories of membrane proteins. There are peripheral proteins. These are not strongly bound to the membrane. They're kind of associated with the membrane surface. Usually we can liberate those from cell membranes just by treatment with a mild detergent. There are integral membrane proteins, and these are like what's depicted here. They're sitting all the way in that lipid bilayer, and in order to liberate those, we actually need to use strong detergents or even organic solvents to get them to be disassociated from the membrane. And then we can also associate proteins with the membrane just by conjugating them to a lipid group. That kind of forms an anchor. That greasy group just like inserts itself into the lipid bilayer and anchors the protein at the membrane surface. And there are a variety of hydrophobic groups that can be attached through various linkages onto proteins. So this shows these three different classes of membrane proteins and how they can be removed. At the top, we see the peripheral protein. In this case, there's just an electrostatic interaction between that membrane protein and that peripheral membrane protein in the membrane. So if we just use high salt, mild detergents, change the pH maybe to alter the electrostatic interaction, we can float that peripheral protein off. At the bottom, we see a lipid-linked protein. And the way we're going to liberate that is we're just going to use enzymatic cleavage of that anchor. One enzyme that could be used would be phospholipase, and that's just going to clip off that lipid anchor and liberate the protein. But in order to release our membrane proteins, we really need to add strong detergent. See that central portion of the protein structure? That's presenting a huge amount of hydrophobic surface area. And we're going to need to put detergent molecules around that region if we're going to retain the three-dimensional structure without denaturing it as we pull it off the membrane. Okay, any questions about liberating membrane proteins? Yeah.
If you use a strong detergent to remove an integral protein, like in the process of removing it, is there a period where like the membrane is like slightly leaking because there's nothing in place of where that protein was?
Often as we do this, as we treat a cell membrane with these detergents, we end up destroying the cell membrane. So the cell is no longer viable, the membrane is no longer intact. We introduce so much damage into the membrane through the action of these detergents that that membrane is destroyed. So we have to have kind of a fine balance between being harsh enough to pull the proteins out of the membrane, but not being so harsh that we denature the protein itself. That's a fine balance and explains why so few membrane proteins have been purified and crystallized.
Is there any mechanism that protects the top and the bottom of the protein? Because it looks like the detergent's only protecting the sides.
So you can see in this depiction here that extracellular side and the cytoplasmic side, those are actually polar groups, right? So they probably don't need detergent, and that I actually like the fact that it only shows detergent as associating with the protein precisely where that protein is buried within the membrane. That's probably fairly accurate. Okay, so on the left here, 3 examples of integral membrane proteins. On the right, there's a peripheral membrane protein and a lipid anchored protein. And you can see in each of these cases, in these schematics, that portion of the protein chain that's in the membrane has got a stable, defined conformation. It's either a single pass of the helix, multiple helical entities all associated with each other, or a beta barrel type structure that's sitting in the cell membrane. So, in fact, these integral membrane proteins are usually either barrels or helical bundles or individual helices. And the question is, why don't we see loops or disordered regions within these membranes the way we would for most other proteins? Any idea? Take a look at the secondary structure content for one of these proteins, and everything that's sitting in the membrane is locked into some kind of repeating secondary structure motif. A portion of the protein that's outside or inside, all these loops, ill-defined region, maybe even some disordered regions of the protein. Any suggestions why? we think about the hydrogen bonding of these chains. Notions about why that would drive this structure formation within the membrane? Yeah.
I mean, the inner membrane, like the lipid section of the membrane, is super hydrophobic, so there's no like there's no interference, and so the chain has to hydrogen bond with itself to satisfy this.
Exactly. The chain has to hydrogen bond with itself because nobody else around there is going to be playing that game. Outside, inside, aqueous environment, you expose a loop or a disordered region, and the amide groups in the backbone, they're going to be able to be hydrogen bonded to solvent water. Within the membrane itself, not going to happen. All those groups want to be hydrogen bonded. The only place that can do that is the hydrogen bond with themselves. That drives secondary structure formation, and that's why you see so much order secondary structure content within these membrane proteins. Okay, so let's begin thinking about the role of these membrane proteins, what they're doing. In some cases, they're just tunnels, essentially. They're just allowing some environment for small molecules, ions, to pass from one side to the other. These are channels. In other cases, the proteins are actually doing work. They're more functioning as pumps. And you can see that as you are looking at the motion, the directed flow of these, let's say, ions depicted here. In the case of passive transport, we're getting directional flow according to the concentration gradient. Whereas in active transport, you can push small molecules, ions, against their concentration gradient, but that's got to be driven by some kind of energy-dependent process. And we'll take a look at what we can use there. This is an example I find really fascinating. It's perhaps the most basic kind of transport phenomenon that the cell needs to deal with, which is it just needs to maintain the proper amount of water within the cell. And it can't count on diffusion alone. As I pointed out, water diffuses, but only at a very slow rate. So there's a bunch of proteins called the aquaporins that are responsible for creating channels that are just the right size, shape to allow passage of water from one side to the other. These turn out to be really important in erythrocytes, in our kidneys, in the renal ducts. Plants obviously care a lot about maintaining water balance inside the cell to outside the cell. And pathologies associated with these aquaporin proteins are associated with a lot of different disease states. So here's a beautiful experiment that proves what aquaporin does. These are xenopus oocytes. These are frog egg cells. They're really big cells. And so we can inject right into these individual cells an mRNA that encodes for one of these aquaporins. The mRNA is expressed, it makes the aquaporin protein, and that aquaporin sits in the cell membrane of these oocytes. Now we begin to manipulate these things, and we put them under osmotic stress. We put them in a hypoosmotic medium in which water is going to want to flow into the cell. In the absence of the aquaporin protein, the cell does very little because water can't flow on its own very readily. But in the presence of the aquaporin, within a period of just a few minutes, this cell has blown up to a huge size under this osmotic stress. In fact, it's been calculated that water can flow through these proteins at a rate up to 10 to the 9 water molecules per second. Not per cell, per protein. Each protein has got 10 to the 9th water molecules flowing through it every second. And that's amazing even when you compare it to some of the most efficient enzymes that are operating at their diffusion rate. Usually these K-cats go up to about 10 to the 7th per second, maybe a little bit faster, and here water molecules are flowing. even faster than that. So this brings up a really interesting problem. Cells need to control concentrations of ions from one side to the other. How do we open up a channel in the membrane that allows water to flow without allowing a whole bunch of other things to flow as well, such as sodium and potassium ions? How do we get that kind of selectivity that only allow water molecules? Well, aquaporin now is one of these membrane proteins for which we do, in fact, have a high-resolution structure. You can take a look at this thing like structure B right here, and you can see right away what portion of the protein structure is embedded within the membrane, because check it out. All of those alpha helices all packed against each other, huge secondary structure content right in the middle of that membrane, and these loops and less ordered regions, they're sticking out one side or the other. Now, at the top, you can see right in the middle of this protein structure, there's a pore. And that pore has got the exact right arrangement to allow water molecules to flow. There are amino acid side chains that stick into the pore that constrict the sides and also provide for exactly the kind of specificity that allows only water molecules to flow. How do we allow water to flow and not other highly charged species such as sodium or potassium? This shows a cartoon of one of the ways in which it's thought that this pore restricts passage only to water molecules. It shows an electrostatic environment that's created by two alpha helical segments pointing their dipoles of the helices right into this pore and creating a positive charge site on one side of the pore. As molecules enter into this region, they actually, when they get in the middle of the protein, they have to turn around and orient the water molecules more negatively polar side towards these alpha helices. And these green arrows on the left indicate this kind of 180-degree flip that the water molecules need to do in order to orient one side of those polar molecules within this channel to the other. Because water molecule has a strong dipole moment, it can undergo this kind of flip and allow passage through, but ions such as sodium and potassium can't undergo this kind of reorientation, so they're restricted from this specificity pore. Okay. Other examples of transport proteins include the erythrocyte glucose transporter. So the cell needs to take up glucose in order to bring it into the metabolic pathways. Concentrations of glucose can vary from outside of the cell to inside of the cell. The cell is going to want to allow passage of glucose, but can't just wait for diffusion. It needs to speed up that process, and this transporter protein speeds up the passage of glucose through the membrane by about 50,000-fold. This was the representation we had for these sorts of membrane proteins not that long ago. We could guess where the protein passed through the membrane just based upon the pattern of hydrophobic and polar residues. But that's the kind of level of structure that we had. Not too much information to go on. We were able to do experiments where we studied how quickly glucose could flow in the presence of varying concentrations of glucose on the extracellular side. And an interesting phenomenon was discovered in which at higher and higher concentrations, at some point, the velocity of glucose entry into the cell became saturated. So when you think about saturation kinetics, what do you think about? So here, maybe this is responsive to the question that I was asked right at the beginning. When you think about this kind of saturation kinetics, what are you thinking about? Enzymes, right. So why do enzymes show this kind of behavior? Right. Because enzymes can have their active sites completely saturated, they're completely bound up with substrate. The enzyme can't go any faster. It's kind of got to wait for the catalytic event to happen and release product. And this is the same kind of phenomenon. As we increase glucose concentration, we hit a maximum velocity. We've occluded all the binding sites. And this suggests there is a distinct binding site and a particular mechanism of transport for this protein. And we have a pretty good idea of what that is. That there is, in fact, a binding event. There is, in fact, a binding site on the extracellular side for glucose. The binding of that sugar group induces a conformational change within the protein that kind of induces a swiveling that exposes a channel from the middle of the membrane bilayer into the intracellular side. And now the glucose molecule has essentially free passage into the interior of the cell. The specificity is really good. That's another enzyme-like characteristic. It binds to glucose, in particular stereochemical form, D-glucose over L-glucose. It likes binding glucose versus mannose. And again, that's telling you that there's a very specific binding pocket within this transporter. It's not just a poorly organized tunnel. So very high rate of diffusion. It goes down the concentration gradient, right? So this is just going along with whatever variation in concentration would normally be established. It's not going against the way it would want to diffuse in any event. So this is not an energy-dependent process. Yeah.
On the graph, 2 slides before, you had the half maximum velocity of location.
Yeah.
Is that
Yeah. So just as we saw with enzymes, when you hit a concentration of substrate that's that gives you the half of the maximum velocity for the enzyme, that gives you information about the Km for that enzyme. And there's an analogous constant for transporters, which is at half maximum velocity, you have Kt, where T stands for transporter. Yeah?
Do all carrier proteins of associated diffusion have this kind of enzyme-like effects?
All the ones that I know about do have this sort of ability to become saturated.
But not the channel proteins?
The channel proteins, like the aquaporin, yeah, there's no distinct binding site that has to wait for a event to happen for the protein to undergo conformational change. Some ion transporters are not saturated, and we'll take a look at some of those in just a second. Yeah?
Isn't the concentration itself of glucose across the membrane that makes the channel close?
Is it a concentration that makes the channel close?
Like as you're taking more glucose into the cell, the difference in the concentrations outside and inside are becoming less. So is that
actually, this kind of motion, this swivel kind of motion, this is a kind of motion that the protein is going to want to be participating in anyway. So many proteins undergo dynamic equilibrium between two conformational states. So I don't think that there's a case that like the variability of the glucose concentration is driving the conformational change. We will see cases in which hydrolysis of ATP is used to drive conformational changes in transporter proteins. Yeah?
This is kind of a review question.
Yeah, go for it.
So for the GLUT proteins that they have different Kn values, right,
Okay.
depending on which tissues they're located in.
Mm-hmm.
But they're listed as Kn, not Kt.
Yeah, so I think all of the examples that we're going to be talking about are true enzyme functions. We won't be talking about any transporter characteristics with regard to what's coming up
So
for
they're
the
two different
midterm.
proteins.
Two different phenomena. Yeah, we'll only be talking about enzyme mechanisms for the midterm. We won't be talking about transport phenomena. Okay. Here's the structure of the glucose transporter, and this just gives you an idea of what these channels look like. This helical element labeled IC3 kind of forms a latch towards the bottom of the protein that can be opened and closed. And just for your future interest, that structure of the glucose transporter was only solved in 2014, and I'm including this passage from the paper that just talks about how truly difficult it was to try to obtain the pure protein, to associate it with just the right detergent. In this case, it was N-nonylbeta-D-glucopyranoside was used as the detergent. And it just must have taken years and years and years of effort to get this protein out of the membrane, stable in presence of detergent and crystallized. Really, really tough work. Yeah.
So if all these facilitated transports have a lot of enzyme-like characteristics, what makes them distinctly different?
Because there's no actual chemical change to the molecule being transported. There's no bonds being.
Okay. Right, they're just going inside.
Yeah. Yeah.
You mentioned earlier about characterization, like destroying the membrane, pretty much totally, but also that some of the lithographs, it's important for them to be stable so that things are associated together. So how would you know if you destroy the full membrane if you're missing some of the elements that might work together with these proteins? Is there a way to isolate a whole part to see how they work together?
Yeah, that's a really great question. And it's true that when you do this kind of thing, like when you treat these things with these detergents, you will oftentimes pull apart whatever protein assemblies have been created. You can liberate intact lipid rafts if you use the, again, right kind of detergents, and you can visualize those isolated lipid rafts. And you can see what proteins are co-localized within those lipid rafts. Again, fairly precise work. And subject to quite a bit of controversy too, I might say, like whether or not you're actually taking a look at artifacts. We mentioned before, when we were talking about structure determination techniques, cryo-electron microscopy. And in those cases, it's like a non-destructive technique. And then you can actually take a look at proteins more within their native context and begin to try to see the way that the protein assemblies might be formed within the membrane itself. All right. Just another cartoon of how these, how the glucose transporter might work and the role of this intracellular helix in forming this latch. So if you're interested in that, you can take a more careful look. Okay, so now let's talk about transport of ions, and this really begins to get us into this issue of cell signaling, because ion transport into the cell is really crucial, certainly in neural signal transduction. So these channels are different from ion transporters that we'll take a look at. They exhibit very fast flux. Again, we can get up to about 10 to the 8th ions per second flowing through these things. You can see where it's sitting in the membrane. All those helices are the portion of the protein that's sitting within the membrane. They can't be saturated in these cases. Again, there's no particular binding site for the ions within this protein. These are more like tunnels. But they are gated, so there's like a toll gate on these tunnels, and these are only partially, these are only open a portion of the time. The cell controls when they're going to be opened and closed, and that makes them ideal for signaling. The signals that can open and close them can include a ligand, like a neurotransmitter, or even the presence of a voltage difference from one side of the cell membrane to the other is sufficient to provide a signal to open and close them. Perhaps the most famous structure that's been solved for an ion channel is for the potassium channel that was solved by Roderick McKinnon. I think that work was done here in New York City, if I remember correctly. So this is what the channels do. They provide a portal that can be opened in response to a signal. And the cell, again, provides for a concentration gradient. And you can see here that there are a lot more ions on one side of the barrier than the other on the left-hand side. And then if we open a channel that allows the passage only of cations, let's say, we're going to change the electrical gradient from one side of the cell to the other. So again, these selective ion transporters can be used to induce an electrical gradient within the cell. Or, once a gradient is established, to diminish the presence of that gradient. So here's an ion channel that's gated by voltage. This is a sodium channel that might sit in the cell membrane of a neuron. And this is going to be responsive to this change in membrane potential. It's sitting there waiting for an electrical signal. And when that signal comes about, the inside of this channel opens up sufficiently to allow sodium to flow into the cell. And that actually serves to depolarize that neuron. That passage of sodium can take place for a brief period of time, and then that channel is inactivated. And it's inactivated through a kind of an unusual mechanism. It's actually kind of a ball-like portion of the protein structure that floats back into that channel and occludes it, just like a cork on a bottle. Here's the structure of the potassium channel. Again, you can see that as the protein is organized, it's got a pore right in the middle of it. And that protein sits within the cell membrane, and here again we need to be concerned with how to get selectivity with regard to the molecules that are flowing through that pore, right? So in this case you see a potassium ion is on the extracellular side, actually on the intracellular side, come into the kind of antechamber of this protein, and then it begins to hit this kind of pore that's sitting within the potassium channel. The water molecules that are surrounding the potassium need to be stripped off, and the bare naked potassium ion is able to go through that pore. So what allows potassium to flow through and not other ions such as sodium? That pore includes carbonyl groups that are pointing into it from the backbone of the protein that are organized just the right way that it can properly solvate potassium ions with these carbonyl groups that are recognizing just the proper size of the potassium ion. And the sodium ion won't be properly solvated by these carbonyl groups just because of the size differential. So really fast flow, but really great selectivity that's organized by the very careful arrangement of groups within these channels. Okay, let's talk now about active transport. So here we're going to expend energy, typically in the form of ATP hydrolysis. And that's going to be used to drive the flow of small molecules against their concentration gradient. And so this can be used in one of two ways, either through primary active transport, where the ATP is being used by that protein transporter directly to drive that, or ATP can be used to drive a concentration gradient, and it's that concentration gradient that then is used to provide the energy for import or export of some secondary species. Here's how these concentration gradients are established. This is the sodium-potassium ATPase. This is an energy-requiring enzyme. This hydrolyzes ATP, and in the process, it brings 3 sodium ions from inside of the cell to the outside. When it exposes that surface to the exterior, it presents a binding site for 2 particular potassium ions, and then it undergoes another swiveling event to allow the potassium ions to go into the inside. So this is kind of interesting because it's creating a couple of things. First of all, it's pushing two different ions in different directions, so increasing the concentration of sodium outside the cell, increasing the concentration of potassium inside the cell, and it's also creating this charge differential. Because if we push 3 positive charges out but only bring 2 positive charges in, this is also creating an electrical gradient at the same time. Now that we've established that gradient, we can use that as a source of energy to transport other solutes. So this is what we call secondary active transport. Here's a list of additional solutes that can be transported, typically imported into the cell. If we want to take up amino acids such as proline or saccharides such as lactose or glucose, we can just bring them into the cell at the same time that we transport one of these ions such as sodium into the cell at the same time. On the right-hand side, it describes symport versus antiport. And so this is one facet that I want you to understand. So if we have a concentration gradient, we can flow 2 units, 2 different species in at the same time, both down their concentration gradient in the same direction. That's called symport. Or we can use a concentration gradient that wants to drive things in one direction as a source of energy to bring another chemical species in the opposite direction. And here's an example of that. This is the lactose transporter. And so in this case, the energy that's providing the import of lactose is established by the fact that the cell maintains a proton pump that takes hydronium and pushes the protons to the outside of the cell. That provides a gradient that can be used to drive the galactoside transporter, and then protons can be brought into the cell down their concentration gradient at the same time that lactose is being brought in as well. Okay? So even though the flow of lactose is endergonic, it's going against its concentration gradient, the force is established by that protein pump. So overall, it's net favorable to bring the lactose in. This cartoon right here shows the different transport processes that are active in the cell: free diffusion, presence of ion channels, primary and secondary active transport that's driven by ATP. So this is going to be a convenient cartoon for you to review when you're thinking about different ways of transport within the cell. All right. So now I want to switch gears and talk about signaling, because in addition to metabolites and ions, we also need to relay information from one side of the cell to the other. And oftentimes, when we need to regulate what's going on in the cell, we want to be able to determine the activity of proteins. So this shows a variety of different ways that proteins are regulated. We can alter the position within the cell, targeting them to particular compartments or associate them with different protein partners. We can bind allosteric regulators onto the proteins to control their activity. We can modify them, for example, with kinases to phosphorylate them to make them active or inactive. We can alter the amount of protein that's present in the cell, either by inducing gene expression or by altering the lifetime of the mRNA that encodes for them. We can decrease protein concentration by targeting proteins for destruction by the proteasome, and obviously all of these effects can be brought to bear at the same time to determine what kinds of proteins are active in the cell at any point in time. So signals can be provided from the exterior of the cell, and they can be used to control these events inside of the cell. And here are some of the signals that cells need to respond to. They need to be told when to grow, so they need to be able to respond to growth factors or hormones. They need to be able to detect things we want to sense, like the presence of mechanical force, heat, odorants, taste molecules. All of these things provide signals that need to be transduced and alter what's going on within the cell. So what I really want to focus on now is what is the structural and functional basis that drives these signaling events. One thing is specificity. And here, maybe our odor response is going to be really a great example. We want to be able to distinguish different small molecules that should induce a very different neural response depending upon very subtle changes in the chemical features of those odorant molecules. So we need receptors that are going to be really specific to recognize one small molecule versus another. We need to be able to amplify signals. This is really true in the case of light, for example, where just an individual photon needs to somehow induce a cascade of events that provide for an electrical signal that goes to our brains. So we need some kind of way to take an individual signal that may create an enzymatic event at one position and relay that into a cascade of events that amplify that signal. We need to be able to have that signal be manifest for a very particular period of time. We don't want to just turn the noise on and just leave it blaring. We need to be able to have that signal be recognized for a distinct period of time and then to shut that response off. And then we also need to be able to integrate different signals. We need to be able to detect conditional responses, like, yes, I do want to respond to the presence of this growth signal, but only if these other events are going on at the same time. So we need to be able to integrate two signals together. The types of proteins that mediate these signals are shown here. I think this is another really effective cartoon to provide an overview. So the classes of proteins that I want you to be able to describe are gated ion channels, which we already talked about a little bit, receptor enzymes that I'll describe in more detail next week, serpentine receptors. These are sometimes known as 7-transmembrane receptors or G-coupled protein receptors, and then steroid receptors, which respond to steroid hormones, which can diffuse into the cell, engage those receptors, and be trafficked to the nucleus, where they turn on the expression of genes. Being able to manipulate these signaling events is really important for controlling disease states. This shows some, not all, some of the critical biochemical pathways that are involved in lung cancer. And you'll see that some of these may be familiar to you, such as the MAPK pathway or the involvement of CDK proteins, the involvement of, let's say, RAS, and within the cell membrane itself, you see that the signals are being initiated by the epidermal growth factor receptor, or EGFR. So there's lots of branches and different kinds of circuitous routes that these signals can flow through. I'm just going to turn your attention to one in the bottom right-hand side, the interaction between beta-catenin and TCF4. And that's shown as driving what's called the Wnt gene expression, which really turns on a bunch of the genes that are involved in cellular growth that promote the growth of lung cancer tumors. The other example that I want you to focus on is the influence of steroid hormone receptors. And this is a depiction of what's happening here. that steroid hormones are capable of diffusing into the cytoplasm where they encounter steroid hormone receptors. Those bind and dimerize, and then they interact with other proteins to form a transcription factor complex that turns on the expression of genes that can either trigger things like proper development, determining sex characteristics, for example, in the case of estrogen or androgen receptors, but oftentimes also control cellular growth in an undesirable way. And both the estrogen receptor and androgen receptor are important steroid hormone receptors that drive the growth of breast and prostate cancer. This shows a little bit more detail on the left-hand side about how steroid hormones can bind their ligands, androgens or estrogens, and then how that induces dimerization of these proteins, association of co-regulatory proteins, and this binding of those hormone receptors onto DNA response elements upstream of genes that they are regulating. So it's here that I want to bring Jeff Schneider in, because his research is really involved in trying to modulate these signaling pathways that are really play such a crucial role in driving cancer growth. So Jeff is a collaborator on some of our projects. He is an MD PhD student, and so I thought I would just begin by having him talk about what that MD PhD training experience is like.
Sure. So hi, I'm Jeff. I'm an MD PhD student at NYU. And so a lot of you might be thinking about medical school, but there's actually more options. And so an MD PhD program is an 8-year program designed for people who want to become a doctor, but also really like science and research. And so medical scientist training program, you go through both medical school and graduate school, and it's starting in science. It's the best way to stay in school forever if you really like school. They pay for your medical school is a huge benefit. So if you really like research, this is the best way to get trained in both becoming a doctor and being a scientist. There's about 40 governmentally funded MD PhD programs all over the country. Shown here. The best way to go about doing that now, if you have interest, there's tons of great research going on at NYU. Go find somebody's lab, email someone like Professor Kirschenbaum, and go work for them and see if you like research, and then think about doing this type of program. So I am a student in the cancer biology portion of the MD PhD program. And basically all of the things that you guys have been talking about now are really important in cancer, particularly the signaling pathways that just got mentioned. They all can combine to drive signals that tell cells early on in life to grow when they should, but unfortunately in cancer to grow when they shouldn't, and to continuously grow and cause disease. Particularly, I study prostate cancer, and the androgen receptor, which is a form of steroid hormone receptor, is basically overactivated in prostate cancer. And it was found 70 years ago, if you deprive men of androgens, basically inhibiting the androgen receptor, you can get decreased growth. And so initially, patients that have aggressive prostate cancer will be treated with anti-androgens, and they'll see a disease regression where their cancer slowly, or their cancer quickly or slowly regresses, and they're healthy for a certain period of time. Unfortunately, cancer cells are evil and smart and can come up with ways to develop resistance to these drugs. And so whether it's via mutations or other biological pathways, things like the androgen receptor can become resistant to drugs that we've found that can target it. And so some of the projects that Professor Kirschenbaum and I work on are developing new therapies for patients that are at this end of the spectrum where their disease is growing despite their treatment, to try and slow growth at that end. And so he mentioned the Wnt signaling pathway, which basically we found and other scientists have found in prostate cancer can play a role with the androgen receptor. So this is a prostate cancer cell nucleus, and this protein beta-catenin binds both the androgen receptor, kind of a co-activator that he was mentioning, and its own canonical binding partner TCF, and they drive drive transcription of sets of genes that cause the cancer to divide. And so our goal is to design a molecule that would target these interactions, decreasing the transcriptional profile, and then either targeting both pathways or one or the other, and then causing the cancer to regress. The exciting part of the research, at least I think, is that we hit a logical end with some traditional small molecule targets. Professor Kirschenbaum's lab develops really new, exciting forms of molecules that can target things that we couldn't think of targeting. So protein-protein interactions are generally considered undruggable, but as we move forward, we might be able to target these and cure some patients. So this is a paper I recently published on the topic in collaboration with Professor Kirschenbaum and Professor Susan Logan. And one thing I want to note is that there's a student on here, Michael Hogborough, who is a chemistry student in Kent's lab that crystallized a really crucial compound a few years ago that's basically critical to this entire project. So as an undergrad, you can really make an impact in the research that's going on throughout the institution and really help going forward in fighting disease. You want to talk about this?
Sure, yeah. The other thing that we're pointing out is that the active form of the androgen receptor as it engages the DNA is as a dimer, and that structure on the left shows how that dimer is organized. And one idea that's occurred to us is to just put ligands onto a molecular scaffold in such a way that we'll be able to bind to both copies of the androgen receptor as it forms a dimer in such a way that we can block the site of association where those two proteins come together. If we can block that dimerization, presumably we can block the formation of the active complex that drives transcription of these genes and block the growth of the prostate tumor.
Yeah.
So that also has proven to be an effective strategy for us. Jeff has also been contributing to that research by using some biophysical techniques to monitor the extent to which those two copies of the androgen receptor are associated.
Yeah, so Kent, you showed a really complicated picture with all these signaling pathways that all cause cancer. And the key is we can learn as much about them as we want to, but if we don't have tools to target them and actually disrupt them in a disease state in a person, then there's nothing we can do about it. And so there's a lot of cool research going on now developing new tools to be able to target these pathways.
And I think the important point here is that if you understand what's going on at the molecular level with these different disease states, not only cancer, it's possible to innovate new forms of matter, new molecules that can modulate these, can interfere with them for therapeutic benefit. So I want to give you guys the opportunity to ask Jeff any questions that you might have, both about his particular research and about his experience as an MD, PhD student. Any questions?
What was your research experience in undergrad? When you go to undergrad, how long did you spend in research that made you desire to pursue it?
Sure. So I went to Harvard College. I was a chemistry major in undergrad. I did research for a couple summers and discovered I really liked research. And actually I had no idea I wanted to be a doctor. And then when I graduated, I found a tech job. So that's a common thing during research. There are lots of labs willing to pay young undergraduate, just recently graduated students to work in their lab for a couple of years. And I ended up in a cancer lab and discovered that I loved cancer research and that it was a really great thing to do. And not only that I liked the research part, but most of the directors of the lab were also physicians, and that I could really make a strong impact as not just a researcher, but also doing the medical side of things.
Jeff, I'm wondering whether or not your understanding about these molecular features allows you to do a better job as a clinician. What's been your experience, or what do you see from your colleagues who've been through an MD, PhD program?
Yeah, I think not only do you have a much better understanding of the cutting edge research of what's going on, so being able to translate things that people discover into practice much more quickly, and being able to understand the papers that are coming out. Medicine is rapidly evolving, and things that you learned in medical school maybe 10 years ago, the professors always talk about this, are no longer relevant. And so understanding early molecular techniques and things like how a dimer inhibiting something could work like that really could help your patients and help you be able to adapt moving forward.
All right. Any other questions for Jeff? Okay, thanks very much. So in both of these cases that we've talked about so far with signaling, we've been talking about the flow of molecules from outside of the cell to the inside of the cell to drive these signals, both in the case of ion channels, where the flow of ions provides the signal, or in this case, where a steroid hormone is capable of diffusing through the membrane and engaging with the receptor in the intracellular region. But oftentimes, the signal engages the cell from the exterior, induces a change in the membrane protein, and it's only that change in the protein structure or activity that's responsible for driving that signal. So that's the case here with these receptor enzymes and also for these serpentine receptors. You'll see that the signal in these cases never actually goes as a signal molecule from the outside of the cell to the inside of the cell. It's just inducing some variation in protein activity on the inside of the cell. So I just want to highlight the fact that in some cases, ion flows can be induced. This is one that's important in neural signal transduction. This is the acetylcholine gated receptor that allows sodium-calcium to flow and provides passages of signals at the synapse. When we're talking about the other cases here with respect to receptor enzymes and serpentine receptors, we'll see cases here where one small molecule binding on the exterior of the cell can be amplified to provide for a lot of activity inside of the cell, multiple enzymes inside of the cell transducing these responses. So the big steps in these signaling circuits is first, a primary messenger must come along and engage a protein target. That signal must be received. The signal then induces some kind of second messenger, and we'll take a look at what those second messengers are within the cell. And then those effectors, those second messengers, must induce some kind of physiological response. Those can be variability of the activity of pumps, channels, enzymes, or transcription factors, and very often leads to variations in gene expression. Okay? So here are some of the second messengers that relay these sorts of information. They can be ions themselves, such as calcium, nucleotides, such as cyclic AMP. And then finally, as we said before, those signals need to be terminated. We can't just leave them on. We're going to talk about some examples of these. I'll probably focus on insulin next week. These three examples all show some kind of extracellular signal, some kind of molecule that engages the cell on the extracellular surface, such as insulin or epidermal growth factor, and then turns on some response within the cell, such as increased glucose uptake or the variability of gene expression within the cell. I'm going to be paying particular attention on Tuesday to the activity of these proteins, G protein coupled receptors. Oftentimes they're called 7 transmembrane receptors because there's 7 helices that extend from one side of the cell membrane to the other. We'll talk about the structures that have been solved, how these engage with the G proteins inside the cell, which are formed of 3 subunits, and the types of mechanisms that are used to turn that signal on and off associated with the hydrolysis of GTP, as we see in this cartoon. So that's a little preview for what we're going to be talking about next week. The ability of a variety of different signals from outside the cell to engage G proteins at the cell surface, induce second messengers that you see here, and then influence events within the cell. A lot of really cool work is being done here in New York. Leslie Boshall at the Rockefeller Institute is working on odorant molecules, and Professor Zucker up at Columbia is working on G protein coupled receptors that allow us to sense sweet molecules. So this is all features different aspects of wiring within the cell that provide signals. And next week, I'm going to demonstrate to you that these wired circuits can be rewired, and we'll have a little bit of a demonstration about how we can influence some of these pathways to screw up the ways that we sense things in the environment around us. So here's our summary for today. Just concluding about membranes, understanding lipid rafts, knowing how membrane proteins are organized within the cell membrane, understanding how membrane proteins can act as transporters, and then beginning this discussion about the 4 types of signaling proteins that we'll finish up next week. And as far as a preview for that, we'll talk about how these G proteins get activated, what they do in their activated states, and how they become inactivated. All right? And with that, I will remind you about the midterm tomorrow at 2 p.m. And I will see you guys then.
Why would you freak yourself out?